Good morning and welcome to our Innovata webinar. My name is Tony Newton. I'm the Vice President of Business Development here at Innovata, and thank you for attending today. This webinar is part of a series that we are planning, given by experts in the field of liquid biopsy and focused on the detection of residual disease and recurrence. As some background, Innovata are a company that were founded by scientists from Cambridge University and Cancer Research UK. Our focus is on high sensitivity liquid biopsy approaches. We have a CAP-clear lab in Research Triangle Park, US, and a highly sensitive clinical test focused on non-small cell lung cancer, currently used by clinicians across the US. Now we're applying our technology platform for residual disease and recurrence detection with a new product we have named Radar. This new product is not the focus of this webinar, but there will be more coming later on that, and certainly we're happy to discuss this directly with potential partners. Next, I would like to introduce Nitzan Rosenfeld. He is the Senior Group Leader at the Cancer Research UK Cambridge Institute and also a co-founder of Innovata. Today, he's going to speak about the many approaches that are being evaluated to detect residual disease and recurrence post-diagnosis and treatment. This is a relatively new area for the application of liquid biopsy and one where sensitivity becomes a major consideration, and here we'll explore some of those aspects. At the end, there'll be an opportunity for Q&A. If there are questions, please send those via the Q&A box at the bottom of the screen. The presentation will run about 40 minutes with some time at the end for Q&A. There's no video in the presentation, and everyone on the call is muted apart from the presenter. And we are recording this for our website, and it will be available later via the website. So with that, I'm going to hand over to Nitzan for the presentation. Thank you, Nitzan.
Thank you, Tony, and good afternoon or good morning, everyone, depending on where you are. Tony, do you hear me well? It's okay?
I hear you, yeah.
Okay, thanks. So thank you, everyone, for joining. I'm happy to speak to you today about our work in the circulating tumor DNA area, focusing primarily in this talk about residual disease, recurrence testing, and very sensitive detection of circulating tumor DNA. As Tony's mentioned, I've been for the last 10.5 years group leader at the Cancer Research UK Cambridge Institute, which is now part of the University of Cambridge, and since 2014, been a co-founder and CSO at Innovata. In today's talk, I want to start with some introduction around ctDNA and its use to guide cancer treatment at different stages and in different places in the cancer pathway. I want to discuss the challenges in general around detecting ctDNA at high sensitivity, some of my thoughts around the best ways to get across that. I'll review some recent approaches for combining multiple markers as ways to increase sensitivity for ctDNA. And then I'll talk about our approach using patient-specific assays as an alternative way to reach great sensitivity for detecting ctDNA by focusing on patient-specific assays, looking at multiple mutations as a way to get multiple markers in one assay. So for treating cancer and for using ctDNA in the cancer pathway, there are multiple places where ctDNA can be used. Basically, whenever there is a clinical decision to be made, the information that clinicians have is always partial, and any further information we can provide can be useful. And one can consider the role of ctDNA in different places in the pathway. Let me just try to get my mouse across here. Where's my mouse? Sorry about that. Where's my mouse? Oh, okay. Here we go. Here we go. All right, so there's many ways in the cancer pathway, from screening or early diagnosis, which is becoming of greater interest in recent times, through molecular profiling, stratification of patients. I'm going to speak later about detecting a residual disease and in other areas during treatment, and it all depends on the technologies we have available. And what's important to keep in mind is that different methods will be useful in different parts and for different types of assays. So it's really important to match the technology and the assay you're using for the clinical use that you intend for this. ctDNA has limitations and challenges. I like to group these into these three groups. One is that there's a small amount of input material. So you're looking at working with a few, maybe 10 nanograms of material that can be extracted from a plasma sample. So we only have a few thousand copies of the DNA, and it's important that we don't lose the material in the processing. Second challenge is that there is a low fraction coming from ctDNA. ctDNA levels can be 10% or even higher of the total cell-free DNA in plasma in patients with advanced cancer. But as you go to earlier cancer stages, and especially if you're looking at patients who have been treated, responding to treatment, and you're going to look for residual disease, then these levels will be much lower, increasing the challenge. The methods we use for analyzing ctDNA, primarily based on PCR and then based on sequencing. And because this is because we are trying to get as much information as we can from the circulating tumor DNA, we're aiming to analyze it across multiple loci in the genome. And as we're using sequencing for that, the errors coming from sequencing start to compound. So it's important that the method you're using is a high fidelity method that reduces any possible sources of noise in the sequencing process. So I'll tell you about different methods that we and others have been using for this, focusing on methods based on amplicon sequencing, whole genome sequencing, and hybrid capture sequencing. And again, how each of these different methods has its role in different, for different purposes and different potential clinical applications. I want to start with looking at the application for molecular stratification or molecular profiling, which is really the first to have entered into clinical use, and to tell you about our work in that area. This started from the assay we described in 2012. This is our tagged amplicon deep sequencing, or TAMP-seq method, where we aim to capture as much as we can from the initial starting material, and through that to sequence multiple regions of the genome by creating a gene panel. And the way we described it in 2012, we started with a pre-amplification to amplify multiple regions in the genome, covering, at the time we had 6 genes in our gene panel. And then we followed that by cleaning up the products and barcoding so that we could perform deep sequencing. In 2012, in the paper by Tim Forshu, Muhammad Murtaza, and our other colleagues, we went as high as 700x sequencing, and this allowed us to detect mutations to about 1% mutant allele fraction. This technology has been taken up by Innovata, which improved it and to enhance TAMP-seq, as described by Davina Gale and others, and validation, analytical validation described by Vincent Plagnol, Tim Forshu, and others in papers from 2018, where we showed the performance of this when we sequence as high as 70,000-fold, and we're now able to detect mutations at high sensitivity, even as low as a few parts per thousand. You can see here some examples. On the left is a time course from the first patient that was treated on the basis of data generated by our assay. This is a patient treated at Gustave Roussy in France, outside Paris. And for this patient, we obtained a sample in April 2015. And by analyzing the sample, we identified a set of mutations, including the gatekeeper T790M mutation in EGFR. In this patient who was previously treated by an EGFR first-generation inhibitor. And at the time, this data was sufficient to allow the clinical investigators to gain access to compassionate access to osimertinib. That was not yet licensed at the time. The patient started treatment with osimertinib in June, and you can see there in July a rapid decrease in many of these mutations in the plasma. So we could track a set of multiple mutations, and you can see them behaving quite in consort for the most part, and you can see additional rounds of increase and decrease as patient underwent additional rounds of treatment. You can also see other features here. For example, you see one P53 mutation that did not appear to behave in the same pattern, and that could be in this instance a case of clonal hematopoiesis. But you can also see that multiple mutations can be tracked down to very, very low levels of really individual mutations in plasma sample. On the right, you see the results of the analytical validation that we published in 2018, and as I mentioned. And you can see next to that on the same graph, analytical validation of additional gene panel assays. You can see that multiple assays can perform well when you are at the level of 1.5% allele fraction. When you go to much lower fractions of ctDNA, these start to separate out, and we're very pleased with the performance of our Envision platform that's able to detect at very appreciable sensitivities, also at parts per thousand, even down to 1 part per thousand there. We follow that through with a clinical validation and a prospective clinical validation that was published last year. I won't go into the details, just to say that we've observed 97.8% concordance with tissue profiling across a 36 gene panel with high sensitivity, especially for these genes which are relevant for treatment of non-small cell lung cancer. And on the basis of this is now being put to clinical use. As part of this clinical validation test, actually, part of the protocol and the funding was provided to perform tissue testing in parallel so that we could perform head-to-head comparison between tissue and plasma testing. And it was interesting to observe that for a significant fraction of patients, tissue was not available for tissue testing, even though the funding was provided. And on the whole, we observed more mutations that were detected in ctDNA compared to tissue, because the plasma was available for all the patients, as opposed to tissue that was unavailable for the majority, not the majority, sorry, but a significant fraction of patients. So to summarize, this is an example of the use of ctDNA in advanced cancer patients, where, as I mentioned, the mutant allele fraction can be quite high. Median levels in different cancer types have been reported at about 10%, but you can see that this ranges over multiple orders of magnitude. And indeed, you do need to be able to detect down to below 1% to catch these mutations in the large fraction of patients. And as you go to earlier stages of cancer, levels decrease down to really few copies per plasma sample that you obtain. And that puts a bigger strain on the sensitivity and on the ability to detect ctDNA. So different approaches would be required in these settings. And a direction for the type of approaches that are required was really demonstrated by this important study by Dennis Low, Alan Chan and colleagues from Hong Kong, who've looked at the early detection of nasopharyngeal carcinoma through screening for Epstein-Barr virus DNA in the plasma. They've had rarely remarkable results through this effort, being able to detect early stage cancers, and they estimated that you would need about 500 molecular markers to achieve this. In this case of the EBV DNA, they were able to achieve that because there were multiple repeats of the target they were looking for and multiple copies of the genome in every cell. But they estimated that if you're going to look for tumor DNA, cancer DNA, you will need to look at about 500 markers. So since then, there's been a range of approaches described where people have been looking at combining multiple markers in different ways to try to increase the sensitivity for detecting early stage cancers. And I've grouped these into genome-wide epigenetic approaches, copy number alterations, and sequence alterations as different modalities through which you can obtain data from ctDNA. Under the epigenetics, I include methylation, which is, of course, a direct measure of the alterations to the tumor DNA, but also fragmentation and representation of the tumor DNA, which are effects of that in terms of how the DNA then gets compacted and processed when it's degraded. Copy number alterations have been investigated by whole genome sequencing, or more recently by an amplicon sequencing approach, which I'll mention. And sequence alterations have been studied by gene panels. And in the latter part of the talk, I'll talk about tumor-informed analysis, which is not suitable for screening, but can be very sensitive for detecting a residual disease, and this is the approach we're taking. The genome-wide epigenetics is an approach that I have to admit, for a long while I tried to avoid that, because it's really based on the tissue specificity and the cell type specificity of the signal in the large part, and not necessarily on the cancer specificity. And the experience of many years of research in other fields, looking at protein markers and others, has shown that this has a lot of limitations. If you look at tissue-specific markers such as PSA or CA1 to 5 in other cancers, you know that these have sensitivity limitations. But it's interesting to note how these markers now, by methylation markers or again representation of fragmentation markers, it's possible to still use a cell type specific signal, but because this can now be defined for different tissues by putting together the list of a long list of markers that are specific to a tissue, this can achieve high sensitivity and specificity, and it's really it's a new type of approach that's not quite like looking at tissue-specific protein markers. An example of this approach is the work by Grail and investigators who are leading these studies for the Circulating Cancer Genome Consortium. As reported earlier this year, the performance that they achieved using a genome-wide methylation and then a wide methylation panel looking at many sites in the genome, more than 100,000, they were able to achieve very high sensitivity for a long list of cancers, including more than 50 cancer types. And especially for a predefined set of cancers, they were able to reach sensitivity of greater than 50% for stage 2, still under 50% for stage 1, but it's very impressive for an assay that's a single assay looking at a large number of cancer types. So I'll in slides next I'll talk about one is multiple approaches and how these can be combined by looking not just at one approach but a combination of approaches, and then I'll finish up by looking at the patient-specific assays and how we're using those to really reach what we feel are unparalleled sensitivity levels. I want to start by mentioning this amplicon sequencing approach that was published recently by Bert Vogelstein and others from the Johns Hopkins group, Chris Deauville as lead author there, where they developed a method to assess unemployees by repetitive element sequencing, they call it RealSeq. And you can look up this paper if you want to see how they do that. I think it's a very interesting approach, but I think it becomes really interesting when they combine this approach together with protein markers and sequence alterations based on their previous CancerSeq method. And what they showed in their paper from PNAS, again going further from their paper in 2018 in Science, is that they can further improve the sensitivity for detecting cancer in plasma by, again, combining these multiple approaches. So this now means 3 different assays done in parallel on these plasma samples, but reaching very impressive sensitivities. And you can see here also on the graph on the right how the addition of these multiple modalities increases the area under curve for this, where interestingly the mutations on their own have the poorest performance. This increases for their set of proteins and is highest for combining all these together. Another very interesting approach of combining different types of data was described recently in a paper by Max Dean, Ashley Zadeh, and their group out at Stanford, where they combine data from their CAPSeq panel looking at point mutations with data that they can obtain from the same sequencing run looking at copy number alterations and other features. And you can see here their sensitivity for non-small cell lung cancers for a high specificity setting was as high as 40% for stage 1 cancers. And if they reduce the specificity to 80%, they can now identify the majority of stage 1 and stage 2 cancer by, again, combining these multiple approaches. Additional approaches described, for example, the Delphi approach by Victor Velculescu and the group at Johns Hopkins, where they combined the analysis of whole genome copy number alterations and the representation of ctDNA fragments in different areas on the genome from a slightly medium level whole genome sequencing there, and a few-fold sequencing of the whole genome. Again, providing pretty good performance there. We also performed a similar type of combination approach where we looked at whole genome copy number alterations and fragmentation patterns. And I'll spend a few slides describing this approach, which we published in 2018. So the approach here is based on the assumption, the evidence that's been accumulated really in the last half a decade, showing that the fragmentation of normal cell DNA and tumor cell DNA when it reaches the plasma is different. And there's been several reports dating as far back as 2015, 2016, by Dennis Lowe and colleagues, Hunter Underhill, Jay Shindura, and others, showing that the fragments coming from tumor DNA are shorter than the fragments coming from the normal DNA. And what we aim to do in our group is to try to use that to enhance the sensitivity for analysis of ctDNA and also for detecting ctDNA in an unbiased way from plasma samples. So what Florent Moulier, Dineka Chandrananda, Anya Piskos, and others in the group did is first they wanted to confirm these findings, and we did this through two approaches. The first one on the left, you can see analysis of samples, plasma samples from a xenograft mouse, where the whole genome sequencing data was aligned to either human DNA or mouse DNA. And you can see that the mouse DNA coming from the xenografted tumor is has shorter fragments compared to the mouse DNA. You can also see very prominently there the 10 base pair periodicity in the fragment sizes. And on the right, we used the patient-specific panel and looked at only these mutations that were these loci which were mutated in the tumor. And you can see there the fragments that contain the mutation are shorter than the fragments not containing these mutations. And I'll get back to these methods later. So to take this further, what Florent and the colleagues did is that they took plasma samples, and you can see here as an example, 2 plasma samples from a patient with advanced ovarian cancer. You can see pre-treatment, you can see a very strong signal of copy number alterations. Post-treatment, the patient's ctDNA levels decrease, and you don't see hardly any signal in a simple shallow whole genome sequencing. But after a simple step of site selection, where many of the normal DNA fragments are removed, you can see a very strong enhancement, and you can see here again a very strong signal for ctDNA presence. And again, you can now identify the same and even additional copy number changes post-treatment as compared to a sample pre-treatment. Dineka Chandrananda took these data and created a new metric for copy number analysis, which we call the TMAD, trim deviation from the mean. And adding to that a set of fragmentation features, she developed a random forest model. And with this random forest model, she created a score, and this score we then evaluated across a set of plasma samples, a large number of samples from ovarian cancer, through our collaboration with James Brenton and additional collaborations, as you can see here. And through this metric, we were able to identify and reach an improving ROC curve. You can see there on the left for high ctDNA cancers, you can see reaching ROC of 0.99 AUC with this combination of TMAD and fragmentation features. And also for ctDNA in cancer patients with a low ctDNA level, such as GBMs, renal cancers, and others, you can see here the increase in the detection of ctDNA by adding together fragmentation features to the copy number alterations. I'd like to shift the focus now to looking at, again, at sequence alterations and how we can do more with sequence alterations. And our interest in developing large panels, patient-specific panels, really developed strongly after our work together with Carlos Caldas and others, where we looked at this particular patient, where we had access to autopsy samples from this patient who passed away as part of a clinical study protocol. Mohammed Murtaza, Sarah Jane Dawson, and the team performed exome sequencing of multiple tumors collected at autopsy, identified hundreds of mutations, and Murtaza developed a multiplexed assay that looked at 200 mutations that each of them was identified to be present in a subset or all of these tumor sites. And you can see there on the bottom 200 mutations slotted on the horizontal, and you can see their representation in the primary tumor, in the lymph node sample, in metastatic tumors in different sites, and then in 9 plasma samples collected over time over this patient's treatment. And what we saw from that is that we could identify not only the recurrence of the tumor and its progression in the plasma, we can also identify which subclones of the tumor were really more represented in the plasma and how that changed. So that gave us really the indication that with a large number of patient-specific loci that are assayed together, we can even identify subclones and small, low levels of ctDNA. And we sought to continue and develop that further. So we developed a program for studying performing tumor-informed analyses. And again, the rationale here is that if we study more targets, then we can collect together more signal, increase the sensitivity. If you think about fixed panels, you can have a very large panel, but if your panel is limited to a small fraction of the exome, then you are going to get only a small number of mutations. And again, previous panels, even sizable panels, reported an order of a few or maybe about 10 mutations per patient that you can track. If you're developing such a panel and you're going to look at many loci, you need to reduce the background noise, and this is something that was important in our approach in order to maintain a low false positive rate. Again, reiterating a bit the rationale and how our thinking goes there. So if you have a plasma sample that has a limited amount of input copies, and one of the constraints and challenges of ctDNA analysis, let's say you have 1,000 copies of your input in your input sample, then your sensitivity will be at best 1 part per 1,000. And the idea is that you're now analyzing, instead of 1,000 molecules, you're looking at n mutations. So let's say you're looking at 1,000 mutations, then now you're looking at 1,000 mutations times 1,000 copies, and now you have 1,000,000 molecules that you can potentially look at. And that increases your sensitivity to be on the range of few molecules per the product of these numbers. And in order, again, to look at hundreds of mutation loci, we start with analyzing the tumor sample, identifying mutations through exome sequencing, and then designing an assay that looks at a large number of mutations in parallel. This led us to really envision the ctDNA detection as a two-dimensional landscape where we have the amount of input material on the horizontal axis and the number of mutations analyzed on the vertical axis. And if you consider the majority of the existing literature, this really looks only at this green shaded region where you have about one or two plasma tubes, so a few mils of plasma, a few thousand copies of the genome, and you're looking at up to a dozen mutations or so. A very large fraction of the current literature just looks at that space, whereas you can increase the sensitivity. The sensitivity is now a function of the product of these two variables, so it goes along these diagonal lines. You can increase your sensitivity by analyzing more material, for example, using plasmapheresis, as has been proposed by a very small number of studies, or you can increase your sensitivity by looking at a larger number of mutations. And this is where we aimed really to look at hundreds or even thousands of mutations, depending on the information you have from the tumor. And this now allows you to get the sensitivity to parts per 100,000, even parts per million. There on the left in the purple shaded region, you can see how we've taken this approach and thought that once you get to these numbers of mutations, you can even analyze droplets of blood, and we've recently published a paper showing that you can analyze droplets of blood by cleaning up this material. This could potentially allow you to reach reasonable sensitivities even with a new type of collection, and I hope that will develop into interesting possibilities in the future. The place where we think this can clinically be meaningful is in the study of residual disease post-treatment. So if you envisage an early stage patient who's undergone, let's say, surgery, and if to the surgeons and radiologists' best evidence, there's no remaining tumor that they can see, you still have two possibilities. The patient may indeed do well, or the patient may relapse. And the thought is that whether we can detect ctDNA can help us predict which patients are going to relapse and have recurrence of their disease. And of course, you can do that immediately post-surgery, but if the patient does not have evidence of disease, you can do that at multiple time points and continue to hopefully detect the disease earlier in its progression, rather than waiting until it's sizable and can be detected by imaging. A growing number of studies have investigated this. I'd like to point a few of the earlier studies from a rapidly increasing literature on this area. A paper by, again, Bert Vogelstein, together with Gibbs and Jeannie Tai and others from 2015, showing in application of ctDNA analysis in early stage colon cancer, showing again the patients where ctDNA is detected post-treatment did poorly and progressed, the majority of them in the first year. The patients where ctDNA was negative in this analysis did much better, although you can also see that about a similar number of cases recurred in this population, although the overall prognosis for that as a population is much better, of course. A similar approach for analysis of localized breast cancer by Nick Turner, Isaac Garcia-Morales, and others at the ICR, where in localized breast cancer, they also reported in 2015 that patients with ctDNA post-initial surgery relapsed within a year, and the patients who were negative post-treatment, post-surgery did much better, although again, especially the patients who recurred more than a year later, many of those were negative in the initial CTD analysis. And both of these studies only looked at a single mutation in the plasma sample, limiting their sensitivity. And indeed, they could detect those patients who relapsed rapidly, but the patients who relapsed later on, majority of those were missed. So in order to improve that, ourselves, along with others in the field, have been adopting an approach to look at multiple mutations in parallel to increase that sensitivity. The TracerX group, UCL, and the Crick Institute, led by Chris Sabosh and Charlie Swanton, reported in 2017 this study where, together with Natera, they analyzed a set of about 20 mutations per patient, identified in multi-region sequencing, and then analyzing those in serial plasma samples from patients with non-small cell lung cancer. And again, they were able to identify these patients ahead of clinical relapse, the median of about 77 days before relapse. Just yesterday, they reported an update of that, but I haven't yet been able to update that into my presentation here. You're welcome to look that up on the ACR website. An approach described last year by Mohammed Murtaza and his colleagues at TGen looked at this approach using a method they developed called TARDIS, where they looked in parallel on either 8 mutations or 16 mutations or more, and they could identify and show that they could reach sensitivities down to about 30 parts per million by looking at 16 targets. And again, showing that with 16 targets, they could do better than with 8 targets. And through this approach, they were able to show that they could detect a much lower level of ctDNA in patients who had a pathologic complete response after neoadjuvant chemotherapy. Turning again to the paper I mentioned earlier by Max Dean and others looking at analysis of non-small cell lung cancer using their CAPSEQ method, they also showed that they could achieve pretty good sensitivity for detecting early stage cancers. But what they did is they went to these cases who were negative using their standardized panel, and they designed, for a subset of these cases, they designed patient-specific assays using the CAPSEQ process. And they found that for half of the patients who are negative with a fixed panel, by designing a patient-specific panel, they could then detect ctDNA down to very low levels, even when these were not detected with a fixed panel. Again, emphasizing the advantages you can get in terms of sensitivity by designing a patient-specific assay. Another recent report on the same type of approach by this group at the Broad, Heather Parsons, Victor Adelsteinson, and others, again, looked at designing a patient-specific panel, and by, again, comparing the sensitivity for a single mutation with digital droplet PCR to the sensitivity they could get analyzing 100 SNVs or 500 SNVs, they could show the increasing power and sensitivity by increasing the number of mutations. And through that could identify a predictive score for recurrence in this population of early stage breast cancer patients. Interestingly, and you can see here that the patients where MRD was detected post-op or at year 1 were the patients where they had a large number of mutations and could get a very good detection limit, whereas the patients who were not detected post-treatment or post-year 1 were the ones where they only had few mutations to analyze and had a much lower detection limit at really few parts or a few parts per 10,000. So emphasizing that for this type of application for detecting residual disease, you really need to get down to the sensitivity levels of parts per 100,000 or even parts per million. So to really get us close to this part per million goal, we developed this approach, which we call integration variant reads, or INVAR. This was developed by Jonathan Wan and Katrin Hyder, two former PhD students in the lab. And in terms of the design, this follows a similar approach where we take tumor genotyping. Using the tumor genotyping data, we design a patient-specific, we identify a list of patient-specific mutations, and we use that to design a patient-specific assay. The patient-specific assay now allows us, when applied to plasma, to get high-depth sequencing across a large number of loci. And we pile all of these reads together in what we call the number of informative reads. And what we do here is, instead of trying to identify individual mutations, we try to identify whether there is any signal across all of these informative reads, which we pile together. And in the optimistic setting or in favorable settings, we can get to as high as millions of reads or hundreds of thousands of reads, but that's not always the case, and that depends on the number of mutations we have, the amount of input, and the efficacy of the process in an individual sample. And we compare the number of mutant reads in the real sample to the mutant reads that we could observe as a result of artifacts in the control reads, and that's our statistical comparator. And we developed various approaches that we've described in a paper we posted last year in bioRxiv, showing how we reduce the background noise there to get to high sensitivity. These include, amongst other steps, read collapsing, using noise filters requiring reads in both the forward and reverse, as well as an outlier suppression approach. You can see here on the right the different levels of noise you can get at different trinucleotide contexts. And you can see this varies quite strongly by more than 2 orders of magnitude, depending on the type of alterations. And some of these can get you down to noise levels below parts per million, which is required to detect ctDNA at these types of levels. Here's an example from a calibration curve that we did by dilution series. And you can see here that by applying all of these error filters together, you can see on the lower curve that even at levels below 10 to the -6, we could still detect 2 out of 3 spiked in samples without getting signal in the unspiked samples. And really supporting our sense that this allows you, with thousands of mutations analyzed together, to get to parts per million and below. Some example data from this when applied to real patients. On the left, you can see early stage melanoma cancer patients after surgery. You can see the detection of ctDNA in the few months after surgery. Patients on the left were not detected. You can see half of these patients indeed did not relapse in 5 years, but of the patients who did relapse, we could detect a good fraction of these, sometimes at levels as low as few parts per million in the plasma. And in the on the right side, you can see patients with a stage 4 melanoma were monitored during treatment, and you can see that we can measure their ctDNA levels spanning 5 or 6 orders of magnitude, from levels as high as 10% to levels of 2 or 3 parts per million in some cases. And these reflect really the presence of less than 1 part per 100 of a cancer cell in a sample that we're measuring. So now we're no longer looking at 1 or 2 copies of the cancer genome, we're looking at fractions of a % of a cancer genome in a sample. I like to use this plot where we're looking at the detection and the levels of ctDNA plotted versus the number of informative reads. And the dotted line gives you a guide to the eye as to the expected sensitivity level, because you cannot detect less than 1 copy in the number of informative reads. And you can see here that in some cases, our detection got to as low as few parts per million. Obviously, that depends on having as many informative reads as in the millions. And in some cases, the number of informative reads was only in the range of a few tens of thousands. Obviously, the sensitivity there is not as high. And this means that for this sample, we know that we didn't have really the required sensitivity. So how to integrate and use that in practice, how to use that knowledge is still something that needs to be worked out. But I think we now have access to that information. Just to wrap up, a couple of examples of how we now are applying this approach to look at cancers of different types. One way is to look at this residual disease setting where we think patient may be cured, but we've also been applying this to look at challenging cancers such as glioblastomas. You can see on the left work by Florent Mouliat with Richard Mayer, Kevin Brindle and others, including some papers that we published last year, also on bioRxiv. And you can see here that using this approach, we're able now to detect ctDNA in the majority of glioblastoma cancer patients, including in their CSF, in their plasma, and in their urine. On the right, you can see results in renal cancer from a collaboration with Grant Stewart, Ellen Heitzer, and Grant Stewart at Cambridge, and Ellen Heitzer and team in Graz. This is a project led by Chris Smith in our group, and he's chosen here to analyze renal cancer, which, if you followed yesterday's AACR, was singled out by the GRAIL group as the a very difficult cancer that they had to exclude. And in this case, again, using this patient-specific approach, we're able to reach high levels of detection in the plasma, but also in the urine of renal cancer patients. And this allows us to study and get some of these features of ctDNA also in these hard-to-study cancers. To summarize, I hope that some of the data I showed today helped explain how ctDNA can guide cancer treatment at different stages. Reminding you, detection of ctDNA at high sensitivity is critical for detecting and monitoring residual disease and recurrence. There are multiple different approaches for analysis of ctDNA. You have to match the tools to your objective and your clinical problem. Also, for high sensitivity detection, there are multiple approaches. And specifically, we've been focusing on patient-specific assays, as we feel that these really provide an unparalleled sensitivity. And this is the basis of what we're looking now for analyzing residual disease and recurrence in clinical research applications. I'd like to thank a lot of people who've taken part in this work in our group, and as well as our close and important collaborators. Some of these names I've shouted out during the talk, and additional ones are listed here. Thank you very much for listening. I'd be happy to take some questions as time permits through our chat box. And with this, I'm handing back to you, Tony. Thank you.
Thank you, Nitzan. And just as a reminder, if you have questions, please send them via the Q&A function at the bottom. We have had a couple of questions, actually. One is, are these personalized panels really practical? Wouldn't it be easier just to go ahead and use a large fixed panel to get the same answer?
Thank you, Tony, and thank you, the audience, for the questions. So for this, as I mentioned, there has been some comparisons between these approaches, and by careful applications of a fixed panel, I think we've seen in approaches such as by GRAIL and also by the CAPSEEK, for example, that one can reach very good sensitivities in applications such as screening. But we've also seen that when this gets very low and these fixed panels start to lose sensitivity, you can improve the sensitivity further by a patient-specific panel. So I think there is, as I mentioned, there is room for both. If you really want to push down on sensitivity, then I think you need the patient-specific approach will provide that extra sensitivity that at the moment we cannot yet obtain with the fixed panel approaches.
And another question is, can these personalized panels identify resistance mutations or changes that take place in the tumor during the course of treatment?
It's a good question. So I think it depends on how you set it up, but it will be with important caveats. So in the setting of a very early stage cancer patient, the way that these patient-specific approaches allow you to detect low levels of ctDNA is by collecting information across multiple targets. So if you have less than one copy of a cancer genome per cell, then even if your assay covers that loci, you have a very high likelihood of not missing, or not capturing that mutation if it's not represented in the sample. I think if your assay, you can set up your assay so that it includes, in addition to patient-specific mutation, also additional mutations that are of interest. But in the setting of very low levels of ctDNA, you're not likely to capture any individual mutations. Rather, you get your sensitivity by pooling together the data for multiple approaches. So I think once you've identified through a patient-specific approach that the ctDNA levels are at levels of parts per thousand or higher, then that would set you up well to use a high sensitivity approach for a high sensitivity gene panel to look for these mutations. But if your ctDNA levels are much below one cancer cell per sample, then you're more likely to miss these mutations even if you apply a stratification panel.
Great, thanks. And then there's another question coming, which is specifically comparing to Guardant or fixed panels. Do you feel that 48 variants, as Innovata are using, are necessary, or could you get away with less than that?
I think this is a continuum. There isn't a hard number that's the right number. I think the clinical evidence is what will determine at the end. As we saw and I demonstrated here and others have demonstrated, when you increase the number of mutations, you have a better sensitivity. I mean, there's no two ways about it. More mutations give you better sensitivity. That's been demonstrated by some of the paper by De Brood, paper by Maxine and others. Also yesterday in the presentation by the TracerX, they trying to improve their sensitivity, they increased the mutations, and I think that also played a role there. What is the required sensitivity for a different clinical application? That needs to be determined. So in general, if you're doing an exploratory approach, I would say use the most sensitive approach you can, and from that you can decide if for the follow-up study you may be okay to reduce your sensitivity. But in case you're not detecting, then probably you can increase your sensitivity by getting more mutations. Whether that would translate into clinically impactful sensitivity, the only way to test that would be the types of clinical studies that are and the clinical trials that are being undertaken nowadays. And I think that's really exciting to see those developing.
Great, thank you. And then on a practical question is, how little plasma can you use for these assays? I mean, because this is often a practical limitation.
Similarly to the previous question, I think there's no hard limit. As I showed you in that graph, and I hope people will kind of get used to using that type of thinking. Your sensitivity is a product of the number of mutations you're looking at times the amount of material. And if you have less material, your sensitivity will suffer as a consequence. We now aim to analyze in the range of a few thousand copies of the genome to start with. So that relates to a few mils of plasma. So if you start with a few mils of plasma and you're looking at a good number of mutations, you can get to sensitivities of parts per 100,000 or even a few parts per million. And if you have less plasma, then your sensitivity will suffer. If you have fewer mutations, or let's say an assay that loses material in the process, then your sensitivity, your analytical sensitivity will suffer through that.
Great, thanks. Another question that's come in is, in a clinical setting for this assay, how can a doctor use a positive ctDNA result without having correlative imaging?
Myself, I'm not a clinician, so I will use that for that, my intuition from speaking to a lot of clinicians through our translational research studies and through attending a lot of talks by others in the field. My sense is that by having this information, the clinicians will at least be alert that this is coming. They could take more conservative approaches, such as increasing the rate of imaging tests, so they can detect the cancer earlier, maybe even providing that as a biased input to the imaging, trying harder to look for evidence. Or if people are more kind of adventurous in their approach, and they do this under an informed consent or in the proper way, that they could perform a trial where they're trying to treat patients on the basis of this evidence. And there are several trials of this type now underway, where within a trial setting, the patients are being treated earlier on the basis of ctDNA detection. And my estimate, based on the evidence presented so far, is that when you have a high fidelity assay with high specificity, if you do detect ctDNA, the likelihood of this patient eventually recurring is high, and therefore treating early is likely to have an impact and to be beneficial for the patient. And so I think molecularly upstaging the patients is going to be an easier thing to start with. Molecularly downstaging patients and telling patients that I think requires more caution and depends on the sensitivity of the assay, because as we've seen with the lower sensitivity assays, there's a good number of patients who do recur even though they're not initially detected by ctDNA. So I think these require the appropriate methods and the clinical trials setting before this is rolled out wider. I think for that, it would be really important to work together between the investigators and pharma companies to conduct such clinical trials, and personally would be happy to take part in such studies.
Great, thank you. And then another practical question is, how long does it take to actually build a patient-specific panel like this?
Our experience from doing this in the academic lab is that we've not applied much pressure to trying to do this at a rapid pace. But our estimate is that this can probably be done in the space of a few weeks if you have a good process put in place to generate the sequencing data from the tumor and then designing the assay and testing the assay. So probably around 4 weeks is an ambitious but maybe achievable estimate.
Great, thank you, Nitzan. That's all the questions for today. So thank you for your presentation. Thank you to the audience for paying attention. If you need more information on the Innovata radar product, please feel free to contact me. My contact is on the screen there. And have a great day. Thank you.
Thank you very much. Thank you, Tony, and good day, everyone.